This is a production of Cornell University.
Thanks, Professor Gan, for nice introduction. So thanks everybody to attend this seminar. So my talk would be today is slightly different from normally like your biology-oriented seminar, but we talking more like the technology-based. So I hope we're using a good example to demonstrate the case how really the new technology or application can help leverage the plant science, particular plant science research. So before I start my talk, I because in past 2 years, the pandemic, a lot of new graduates in the new faculty may not know who we are, what we are doing. So I try to get very brief to tell everybody what we are doing. So basically, we're located the first floor of biotechnology building. It's just and we have current five staff member, and we have mainly we have one position open for tech position for metabolomics side, but mainly we the key is we have the tool called mass spectrometry. And that's the key part of the tool, very expensive one, but can do a lot and facilitate research, a lot of things that we are going to talk about today. And obviously by informatics tool here, and this is the help us to be able to do the data analysis. So this is basically a service a lot of people probably don't know. I really encourage everybody, if you get a chance to look at the website, we list for all. And this is the proteomics side. The main thing is today we will talk about the major things, but one of them, a lot of people were using called protein-protein interaction, protein-ligand interaction. We normally use an IP down and the cross-linking mass spec. So I just mentioned that. And untargeted metabolomics side, other than today we talk about, we're doing untargeted lipidomics, and then we're doing quite a bit is a target. Once you know your molecule or the compound you are looking for, you're trying to do absolutely or profiling, then we're using targeted approach to do analysis. So as I said, today we're going to talk about really the three different types of technology or workflow, and just the very past 2 years developed. And one cover with for proteomics, two all related to metabolomics. And then I will show each of the case done here. What's the technology detail about, and obviously we have a three cases to demonstrate. One of the cases is, it's called the new technology name called real-time search, MS-3. So a lot of biology people probably don't know what the MS-3 mean. I think I will explain a little bit late at this point. But the technology coming out in 2019, the summer of 2019, it's called, this is a dramatically improve the quantitative proteomics, which is most widely used application for proteomics side. So one of the, this is mainly the improve is the outcome would be increased significant accuracy and really throughput, which is really help improve the proteome coverage. So we talked about it. By that time, the summer of 2019, we have a project that is coming just from the hydrogen sulfide that treated the lung of pigs. We're trying to, and we just acquired the data using the traditional way, and then this is the perfect time. We got the new equipment, the new technology, and we want to test. And this is how our facility postdoc and Qing would be, deliver the test results. And so I was able to show. So we just mentioned what's the technology about is for quantitative proteomics. But so really, one of the quantitative proteomics we use is called the TMT tag, isobaric tag labeling approach. And the tandem, the TMT tag, the full name called tandem mass tag. And the biggest advantage of this technology is they can do multiple plex. Currently, up to 2 years ago, they can deliver, they can deliver so-called 16 sample. You put it together, you can quantify at the same time. Actually, 2 months ago, almost 3 months ago, now they deliver 18. So we can now doing 18 plex together. So to just make a long story short, a little bit to tell what's that technology about. The beauty of that technology is the reagent. It's called the isobaric reagent. That reagent, they put the reporter ion here using different isotope labeled, and using the balance group to balance the mass. And therefore, that's what they call the isobaric mass. They have 16 or 18 different reagents, but their mass isobaric would be the same. Okay. So when you do, this is 10 years ago, I did for also plant biology. This is department, it's stress, the joy stress, the mulberry. And by then we only did a 6 class. And then once you label the reagent with the peptide level label, and when you do mass spec analysis, in the surface scan, we call it when just the measure peptide, they actually add the same peptide from 6 sample or 16, 18 sample, they all added together, actually boost your signal. But when you do MS2, what we call the MS2 sequencer, when you start sequence or MS3 sequencer, then you get it phlegm ion, the reporter ion relieved because the balance ion gone, and then released, then you are able to do quantitation. So that's why this technology allows you simultaneously identification and quantitation. Now, now the problem, the downside of traditional technology this way is we call it MS2 quantitation. We select peptide and sequence it, and then get a quantitation. The problem is they call it ratio compression. The accuracy significantly compromised. Why they compromise? Because any ion, when we select a peptide, what we call the ion in mass spec, so you're always isolated in some background ion. So for identification, no problem, because this is the dominant. You can, identify what that peptide. But for quantitation, they add all background other ion. So that's why the ratio is not accurate. They call the ratio compression, sometimes called distortion. Okay, so this was really published 10 years ago. So people coming out, a solution, how we address that, because we know the ratio we coming out is not as accurate as we hope. What we're going to do, what they're going to do is mass spec, they can do multiple stage fragmentation. That's MS, well, here we call it MS to the third. So the way you do it, you isolate your peptide you want to quantify after labeling, and then you do fragment. And after the fragment, instead of directly measure the quantitation, then you go to the next level, so-called MS to the third. Select the product ion to go to the MS third. Then you look at the quantitation. Obviously, your background a lot of low. So, but generally speaking, by doing that, then you add a problem is the low sensitivity because you select one. So people developed called single chop precursor selection, SPS. You can select up to 20 now on precursor ion. Normally people do 10 and then boost sensitivity for the quantitation. So that's you're getting away with it. So the ratio obviously now you are a lot better. You can see it down here. And MS2 original was 10, now become 4. But when you do the MS to the third, it's almost getting the real no interference on ratio. So, but any new technology you develop, you're getting benefit on one side, you create another one. And that's one of the typical example we want to show. But you see, this is otherwise we call the MS third is great. The mass accuracy is now solved. But the problem is created. Another problem is because you need additional scan time to do MS third. So what happened was your accuracy improved, but your number, what we call the proteome coverage, now you drop to only 65%. If you have, let's say 80,000, and this is probably only 5,000 you can get. So people don't like that, of course, but Now you have to compromise. You either take the high accuracy or you take the low coverage. Now, so that's one of the technology latest, about 2 years ago, sorry, they discovered they called real-time search. So this is the real-time search. The reason that, well, they basically saying we can speed up the MS to the third originally do by doing real-time search. What that means when we retrospectively look at the original data, we'll find that a lot of MS3 we spend the time on it, but is no quantitation information. Is we call it's unproductive spectral. Therefore, now when you do real-time search, when you put the real-time, you know this has to be the real peptide. Then you do MS to the third. So by doing that way, we really speed up and offset the original we call the time, addition time you need to take. And then this is coming to we can look at it come to see how much we close the gap. So original approach we using is object fusion. We got it, but now the new technology called object eclipse, we are able to do that. So now here's the results. Obviously, in our lab, we also optimize those parameters. There's a lot of parameter. One parameter we optimize using isolation window. So after we optimize that isolation window, I'm not trying to give too much detail. So after we all normalize, you can see this is the original fusion we're using regular SPMS3. We can close the gap to the 75%. The original, most of the people was 65 or 70, little bit. But then now using the latest, the real-time search, everybody can see it. Obviously, this is new machine is a lot better than older generation. It's getting the more, but it's close to almost 92%. The gap original from 65 now to 92%. That's why we get the deeper income, but also data quality, you can see it. The data quality a lot better in terms of ratio complexion, the accuracy. The heat map, you can show a lot of, this is the data a lot reflected actually the abundance change in the real biological sample. So because of that, we did, we're using that, as I said before, the project as a demonstration. So we just acquired using real-time search to find out by doing real-time search for the quantitation, we find additional so-called novel biological pathway involved the cell apoptosis. So, and obviously the graduate student who submit the sample, they test a couple of that pathway and they use internal assay, obviously, to look at it and compare the two different groups to look at it if there is a broken down the DNA fragment and it has. And he also did the pathway others that apoptosis pathway. They also several mark gene. He did the RT-PCR and to demonstrate it is increased by the hydrogen sulfide treated group. And then obviously he also did a West blot analysis to confirm some of the protein change. So after we did that, we did a lot of plan because this is a demo case of the technology that works well in our facility. We now exclude using that. And then Professor Gang has a project coming out. We did the plan. A lot of plan work has not been published, but Professor Gang is waiting to share their data. So I'm happy. So now I just show him, this is the work we have been done for Professor Gang's lab. This is a Professor lab is focused on investigating the regular mechanism of liver synthesis. This is the sample we did was parallel proteomics and phosphoproteomics for liver using rhabdosis as a model system. So what their lab find is they find that knockout a gene called the senescence associated gene 114, which is really a novel protein kinase. What they find was really accelerate the liver senescence. And that's interesting. That's why the project created by here is that where we're trying to identify what's the target protein targeted by SAG or phosphorylated by SAG and related the liver senescence. Okay. So in the real-time experiment design, we actually designed, using 3 different liver stages. I believe this is NS is is the DAG data after germination, 40 days, 45 days, and 49 days. And then we designed with two different wild type versus mutant, actually TMT 16-plex. But down here, we just want to quickly show the wild type results, and then just simple. So the way we just for simplify the case to demonstrate how that works. So the once you TMT label, you put together, I mean, obviously it replicates, and 90%, more than 90% we use for enrichment for phosphoproteomic analysis, and then only 10% we're using global. That's what we use in real-time search, TS. So here's the results, and this is quite an impressive result. So we got a global like almost more than 6,000. That's routine. The more important thing is that we find that Professor Gans lab find more than 200 new senescence associated gene all correspond to the protein, obviously. And from phosphoproteomic side perspective, also find 450 new sites, and that's also very impressive. And here, when you look at the principal component analysis, those three different stages is really separated very well, and data show quite reproducible. And obviously, everybody will do the motif analysis, and you can find, and there's two top, MAP kinase and the casein kinase too. Those are become the top kinase motifs we find for the peptide we identify. And this is everybody know MAKP is responsible for cellular response, in particular in a broad array of the stress. Okay, so that's pretty much we show this case. So the take-home message for this one is I hope to convince you, well, at least we show the real-time such a close gap from original predicted 65 to 70 or something like that, we get more than 90%. And the second point is very important that this is not only we improve the gap, but really accuracy. And that accuracy is very critical to make it possible even confident detect small change. Because in biological, originally, because racial complexion, we have to look at, okay, above certain level, but now allowed us, we routinely can, as long as your data replicates the data and statistics show significant change, even now can down to 1.2, 1.3 for the change, you still see the significant difference. So that's the point I want to make for that new technology. Now I want to switch the gear to the metabolomic side. So in our facility, we recently focused on, the last year, we focused on generated a so-called spectral building in-house compound spectral library. So in this talk, we are trying to demonstrate how this is the library really help facilitate untargeted metabolomics, and we're using plant as an example. And this work was done by the two Lachika and in the facility. They did the enormous work, and to get where they are, this is really helpful. So everybody probably will ask why we need building library. So for untargeted metabolomic, we're always, again, the same. We're trying to identify, screen out what's the difference between the two cell state, or healthy white or mutant white type. Okay, so the way originally metabolomic, we basically copy like a proteomic. We're using public database, which is available with doing search, we find, and then identify what the compound show up. But turn out, it's actually a lot of false positive identification, or called annotation. And the reason why is because public database no rotation time. That's a very important point. So because on those days, our mass spec is very accurate. We can down to 1 ppm or just less than $2 million difference. So we can accurately measure the compound, and even for MS1, MS2 level, the fragment. The problem is that for identification, this is, so we can generate a very good, accurate chemical formula for that compound. But in terms of name identification, often we give high percentage, increase more than the high percentage rate could be long name. And this is the bigger problem because our biologists that we based on the compound names, then we do pathway analysis. So this is where lead high false rate for generating the really for not a true biological hypothesis. So that's why in the past 2 or 3 years, our field of untargeted metabolomic, strong suggested everybody, trying to do untargeted metabolomic, ideally you created your in-house library. So that's what we did because that's the, so with that, so here is just to demonstrate another example why the library is important. The 2 amino acids they done here, one is a valine, one is a aine. They are structurally obviously quite different, but chemical formula same. Fragmentation-wise, they are very similar. Sometimes, often to miss a call between of them. But when you look at the rotation time, they are very different because this is much more hydrophilic in C18, obviously. And we have also lessons in our facility. We did, 2 years ago, 1 year ago, we did for untargeted metabolomic for glutamine. This is the amino acid that we find using, this is a dog sample on health versus disease, okay? So without our own library, when we search, we find the 2 peaks. One is 101, one is 189.5. That's what the identification. The worst scenario is the quantitation change is opposite. The second peak is high in healthy, the first peak is high in disease. So what are we going to do? So now we have a library. When we look at the library, it should be the first one is correct one, the second one we just cross out. So that's just how important the demonstrate the case. So otherwise you will confuse the PIs. So now we build up almost 1,200 total spectral library and catalog it because some overlap. Okay, so I think. Okay. So really, the way we do now here, we add another level, well, we call the mass accuracy for scan and the fragment, and then add the rotation time. That's really boosts our confident identification or annotation for the metabolite. So the benefit is not only now using the library, we can help, ongoing the project, but more importantly, we think we can retrospectively research against the older data. Now we can see how much improved. And obviously that will also help the other targeted analysis for method development. I'm not going to touch here. So one of the projects that we demonstrated here is from, Professor Lab, Greg Peck. That's 3 years ago we did the work. No library, so I will show. They really want to, trying to look at the metabolic analysis for 15 different cultivar on the apple gels, and they're really trying to know what's the composition change or what they are in terms of polyphenol composition. And obviously, the goal they're trying to do is identify those apple contain the sufficient endogenous polyphenol for industrial scale. Then obviously you don't need to add for some of the apple sides. For certain, you don't add additives. That's always the best. So Greg group already did the analysis at the beginning. That's what the slide that he showed me. So it's just 2 cultivar apple side that they use, and you can look at a lot of difference in terms of composition. The abundancy by the polyphenol, one of the pro-N-cytidine, obviously, particularly one of the chlorogenic, the rest is flavonoids. But of course, Greg did only 10, and obviously he trying to get in more information. So we did it was 3 years ago. That's 2018, I remember. So that's triplicated on 15, and then go to our routine way to do the analysis and to get the untargeted profiling metabolites. We did the initial search, and now we just go back to using our own library research and see what do we get. Okay, what is the difference? So here we go. This is the 4,000 feature we identified in those samples, 45, total 45 sample. And the June 2018, that's what the only public database available. We identified 300 annotated, 328 compound, and 49 was polyphenol compound. Now, just a few months ago, La Chica did the research, so we find the 414. And now we find 102 compound. And the reason why, because you can see in terms of number, 60% now the total number gain is polyphenol. And that's because our now library including quite a bit of flavonoids and the polyphenol compound, and that's not a total surprise to us. Okay, so here I just quickly show up some of the compound. This is a polyphenol, and you can see it, in the 15 apple side, they are very different in terms of abundancy. So you can see, I just show those two polyphenol down here first. And then I show a couple, you see, one important thing I show. Yeah, so principal component analysis, we can see, this is the original 49 polyphenol, and you can see they are really 15 cultivar, they belong to 3 groups. And then now we research the 3 group, slight change, but overall was the same. And QC is a mixture altogether. You see our data analysis is really relatively reproducible. We can't. And then Greg already told me, Collar and Zepata, they both are share very close genomic background, and that's what exactly we find here as well. So, I want to go to quickly, and then again, this is a show. It's called gallic acid, and this was belong to the 3 group. There's the 1 group we can identify those 3 cultivar 3 group, but the rest are the same. And the next one next one we just show both caffeic acid and another flavonoid compounds. Those are accumulated or more in those 2 color and the Zepata. That's so you can actually define that which species or which cultivar contained more. So to summarize what this project is we did was we just want to show the in-house spectral, how important the help, not only ongoing project, it's very important that can actually retrospectively help all the data we acquired with the current and the library we build up, as long as we're using the same condition to get the data. So that's, I just want to send the message out. So I hope last 15 minutes or 10, 15 minutes, I want to show another new technique or technology we implemented or developed in our facility, which is really help. And this is technology, the name called a substructure-based profiling and annotation workflow for high throughput identification of novel tomato flavonoids. So now we're talking about the flavonoids. So this is a project we got lucky working with Professor Johnson and Lowe's lab. I know there's a long-standing interest in Johnson group, but they're working on cuticle research. And then we know tomato is an excellent model for cuticle research because they have a very good published annotated genome sequence and the other genomic tool available. More importantly, because they have a very thick, tomato has a very thick, remarkably thick, cuticle. And then this is just to show the across the section of the fluid that you can see. And this is obviously cuticle being stained by red color, and so that you can highlight that how thick. And then this is what we call this is the picture just to show the cuticle is naturally orange color because of contained a lot of flavonoids, particularly Norwegian chacon. Okay. So this is a project that also interesting. They kind of cuticle represent the extracellular components for flavonoids, and this is the epidemic side that we really represent the intracell components. So how we can find the difference. Okay. So now For the flavonoids, everybody knew is a plant second metabolite, right? So chemically, it contains 15 carbon scalene with 2 phenyl ring and connected by hydroxycycline in the middle. So that's called ABC. And the structure very diversity. And the main factor that contributed diversity is because the glycol conjugates. Because a lot of different sugar put on with a different position and a different type of sugar and how different number of the sugar. That's why. So everybody knew flavonoids is very important components to our health, like our diet components to our health animals. But also more important for everybody, they have very good biological function, right? So, but for here is the pathway. Flavonoids, since it's a pathway, which is the most important enzyme down here is called the Chacon synthesis down here. And we highlighted here is the red called CHS. Okay, remember we were talking more. So in tomato, the gene, the CHS gene was well characterized. There's 2 of them, Chacon synthesis gene. And the silence, both genes will produce the purple fluid instead of orange, because you completely knock out the orange color narraging Chacon and the flavonoids. So that's the observation. So Johnson group, this is a working on, obviously this is a gene expression. This is a gene expression interface, what we call the expression cube, with top 10 co-expression the gene, along with the important one we already signed the CHS2 at the first step. Okay, so this is a cube really show a 3D map, a heat map, and of the gene expression across. This is a in one dimension across different tissue from seeds to outer epidermic, and then in the different development stage in the second dimension. And the head part in the third dimension is here is really the gene included the 6 of them important related for a person along with CHS2. And then, oops, here. And then along with the one, this is a transpiration factor actually in regulation all those, some of the pathway genes. So to make a long story short, those are the 10, those are the top 10. Then we already find 7 of the top 10, those genes hit from 35,000 genes was functionally related, flavonoid biosynthesis. So this is the proteomic transcriptomic data. So to complete the transcriptomic data, so what we're trying to do here is we're trying to answer how many flavonoids we can annotate, identify from both the cuticle or epidermal cell. Well, later on we talk. And more importantly, we also know what's the distribution between cuticle and the PO sample. We will touch that in terms of this project. And then finally, also very important is what's the mechanism, what particular traffic mechanism of the flavonoid mechanism, trafficking mechanism between cuticle and the epidermal cell. So we started with that question and we designed the experiment, obviously. So Johnson group did, this is the white type, well, PO sample really including both intracellular cell and flavonoids and secreted, really included both. And he did using CRISPR-Cas9 knock out the 3 mutant gene. And we did 5 replicates, and same things for the sample using critical. And we normalized it by the same weight, the weight same. And this is how we did start from 40 sample. Now, to analyze the flavonoids, there's always a challenge because we already knew the majority flavonoids is glycol conjugates, right? Majority are glycol. So there's obviously no really standard compound available. And because of glycosidic, it's very weak bound in mass spec. So in MS2, when you, this is a loading, when you, just to show you example, when you add MS2, you give you 2 peak. There's no further fragmentation. Therefore, you cannot really identify, you're not sure whether this is a flavonoids or not. So that's why we need additional level MS to the third. Then for the fragment, the cost structure, then we know this is definitely quercetin. Okay, so we know. And then again, if you, this ion, if you do MS, they just give you lots of additional water, additional sugar, and then give you the quercetin. And you need a MS to the 4 because I cannot draw, but I just draw this side, MS to the 4. Then you know for sure this is the structure, how we identify. So we take advantage of our, we have an instrument that this is a 2015, well, 60, now almost 60 years ago we got, and this is called a tribrid instrument with very high accuracy and allowed us to do really, well, they claim up to 10, MS to the 10, but practically maximum we really can to do MS to the 5 in terms of sensitivity we can see. So we take advantage of that, allowed us to facilitate structural eludation and identification isomeric. So the compound, okay, so here's the way how we did. So the way we did it was what we call the MSN based. That means we have to go to MS3, MS4, okay, for identification for flavonoids. If it turns out that in the sample contained only those cost structure and we have a database when we search, we already knew, okay, this is a known annotate. Let's see, sorry. This is driving me crazy. Yeah, okay. So, okay. So the previous, if you already have a database, they actually directly identify what they are. But most of the case, like when you're MS2, you couldn't identify, like this is the compound. And then you go to the MS to the third, okay, now you find that this is a cost structure 317. And the 317, when you do database, okay, we find that this is called isohamnidine down here. And this is clearly flavonoids. Because of that, then we know, okay, this compound 641 has to be flavonoids because that's just because they contain the flavonoid cost structure. So we identify that analysis by identify flavonoids class first. So this is a 2 data analysis process I want to quickly, this is more technical. The first step we use in software. The software allowed us to analyze those, it's called MSN tree data. You have 1, 2, 3, 4, or even 5. You have tree data. Normally the software you couldn't do it, but Mass Frontier allowed you to do. So then we can, by doing that search, we know, okay, those are the compounds contain the core glycan structure of flavonoids. We know those are what we call the flavonoids class compound. After that, then we put a regular like a database search called compound discover. We can annotate, we can identify what they are. So let's see, next step. Okay. So now if we turn out we identify those compounds in compound discover or in Mass Frontier, there's a way, this is called, like we do in routine, called fragment ion search. So how we know what's that? We base on mass spec fragment ion, see if they match the structure. So the match structure they call a fish. They just take the first word called a fish search for ranking structure. For example, this is because remember the flavonoid compound contain a lot of sugar and they have a lot of isobaric. Their mass basically exactly the same. So how you determine which one is which? Okay, so the way you rely on MS to the 3 or MS to the 4. You see, this is the structure kinfolk. This is structure down here. The MS3 down here support. This is the flavonoid. Okay, it's a kinfolk of flavonoid because 3 hydroxyl group here. But this one actually is not this one that come out early. This one actually flavone because there's no hydroxyl group here. So clearly MS to the 3rd or MS to the 4th will clearly tell us which one is better. And the way we don't need it manually, we just using fish fragment and match which one is best to us. So overall, to summarize, we have found 562 flavonoids in those samples. Obviously, the difference is pure and the cuticle, which we expected. There's quite a difference. But regarding the transporter versus the white type, we really didn't find not much difference. But one of the things that I want to talk about how we identify those unknown. This is what we call a putative structure of a novel. In other words, there's no public available. This is a novel compound we identified from Justin's sample. This is typical when we do the database search happening. This is a substructure available in database. And once it's available, we know, okay, we find that this has to be, this is the major structure down here is from here. This is substructure. But now the mass didn't match, right? Then the mass is a lot more. We have to have a 278. The compound is here. So what I did was basically I do the 278, and based on fragmentation MS2, MS3, I know there's pentose. There's another pentose that should be here. And then I come out of the mass, it has to be additional 146 or 367. Our mass, as I said, very accurate, cannot be wrong. And that fitted is what that is. This is a chimeric acid. And that chimeric acid binds here, and then the mass, everything would perfectly fit our fragmentation. And this is the one. Recently, is the most exciting we find that, well, for this project, in my view, is we actually identified, well, a putative amino acid conjugate. And not really much, well, I didn't see it, the report on this one. And we find the 3 different forms. I just want to use this one as an example. It's an amino that currently we believe is cysteine. It's called the isobond down here. And this give us perfectly fit for all MS2, MS3, MS4 in multiple form we hit beautifully. This is one of the, so obviously this is, we're going to try to, well, we plan to do the NMR to validate that structure. And then they're only presented in epitomic cell, in pure sample, but absolutely not in cuticle at all. So this is the overall, just the summary of the quantitation. So quantitation is basically pure sample. You can see those are the nitrogen is dominant in pure sample, but nitrogen chacon way dominant. Is like quercetin, malicetin, and camphor way dominant in the cuticle, which is those are, nitrogen chacon is a lot strong in terms of antioxidation than nitrogen. That makes sense, in cuticle. They have a much stronger, seems in terms of flavonoids composition, there's much high, strong oxidation feature. So to summarize what we get for this project, we identified total 562, and the Justin told me, well, more than 300 in tomato was new, but in the total database we searched, we find the total 75 was normal. Yeah, we cannot hit any existing database. And we already kind of indetermined 16 putative structure. Interestingly for Justin, we find that 26 out of those are much higher the level in cuticle than in pure sample. In other words, in extracellular, much higher than intracellular, and those are the compositions. So we highlighted here the red, means the high in the pure sample, and then orange was in cuticle sample. So we kind of answered the question what and where, and obviously the important question is, what's the traffic mechanism? And if there is any transport allowed responsible for specific flavonoids transport from epidermic cell to cuticle. And this is, we hope the data, we dig deeper and we can figure out what it is on this. And as I said, whether those processes are, well, so All right, so and then a lot of questions you can ask whether those trafficking mechanism related enzymatic reaction or not, how they produce it in what's it called? How they formed in the epithelial cell and then pump out to cuticle, or whether the how that function doing, or we still need to figure out. So the last that I want to obviously not least is acknowledge those the lab we present the data in here, and the more important I want to acknowledge those two, Henning brain or the new equipment that allowed us to do a lot of work now. This is the latest one for real-time search and through HHMI. This is our NIH grant. I also want to particularly mention Frank Schroeder, BTI, and Ted and has at USDA. They provide as a gift for the standard compound, allowed us to do the data analysis, to making the library. So with that, I want to thank you everybody for your attention, and running 5 minutes late with sorry. So if you have any question, I'd like to answer any question you might have. Thank you, Dr. Zhang, and for excellent. I'm really impressed that there's something user data actually identifies so many actually novel compounds or genes, as Dr. Zhang showed here. Actually, so if you think seriously about your project, you should be able to just contact Dr. Zhang. Any questions? Well, any question from the audience? You should be able to see it. Let's see. It's here in the chat. Oh, great talk. Great talk from Jim. Okay. So if not, I have a question actually about your first RTAs. Actually, the RTAs improvement is it because of software improvement or just technical improvement? Because it seems to me the tandem spectrum 3 times, right? Mass, and mass. Both of them, for example, compare with SPAs, the previous MS3 actually. So essentially the same, but the improvement is the real time is because of software improvement or really technically improved something? Mainly software. This is a very good question, Professor Ken. Technically, we can running. This is the real-time search in object fusion as long as they give us the software. So really, software can control real-time search. There's a little bit related to the hardware because now the new instruments are getting fast. So in other words, the scan rate, everything fast, like I've showed data, because the latest instrument is going to be something around 10 to 15 more, because that's what they fast. So the answer is yes, you can use in any instrument as long as they have that type, can do a message to the third, you can do it. Like a Haywan's machine also you can do it, but the problem is the company we want to sell the latest instrument, they won't let you do it. So in other words, now we dedicate only that instrument to the software only in the latest instrument, the older instrument we can do. But that didn't bother us because we can use that instrument for small molecule for other purpose, like we send, like for flavonoid identification. So that's the software is the main thing. That's great. Thank you. Yeah. Any other questions? Yeah.
With MS and where you're maybe going up to MS to the 5th or whatever, how many MS1 peaks can you target that are coming off at the same time?
Oh, so those are MSN. Because the machine is very fast, right? Acquisition. The way they do it, they select I didn't show that slide, but you selected one precursor ion, the major, and you flag MS2, MS3. So the way that the machine is, once if you set the MS to the 5, they will do all the way to MS5. Obviously, the reason why I said MS practically MS5 is not, MS5 is still okay, MS6 or to the MS10 wouldn't work because it's just no sensitivity. Because once you select at one level, your sensitivity drop by almost tenfold. So that's why. Okay, so to answer your question, so they do from MS1, MS2, MS3, MS4. They didn't finish one first and then select the next one. As long as your machine set the cycle, you want the machine set the cycle 3 seconds or 4 seconds. So that, well, usually less than 3 seconds because you want to also doing quantitation at the same time. So in order to do quantitation, you really need enough data point, what we call the survey scan in MS1. So that's why you compromise this. You might have missed something low abundant ion because of doing that. So the way machine setup is, if you set the MS to the N, MS3 or MS to the 5, we normally set only up to MS to the 5. So they just all the way to finish one thing. As long as above your threshold, you set up. So they will do finish this one and then go to next. Did I answer your question?
Yeah, so it sounds like it does take maybe a couple seconds.
Yes.
And so if you have pretty narrow peaks that are maybe 5 or so seconds, you're somewhat limited there in terms of maybe just targeting one at a time.
Yeah, that's a good question. Yeah, if for what you're talking about is you're trying to target some, you already knew you are interested, right? Trying further categorize. For example, if I have a compound, I just make up 985. This is the compound M plus H is there. So I want to categorize. If that's the case, you can just set up a target. I even don't look at the rest. I just target that one. If you have one. What I'm talking about today is we're trying to screen. We're trying to find anything. They are belong the flavonoids. So we're using the MSN to fish out what they are, whether it's flavonoids or not, and what the change. So that's a more complex way we do it. I hope I answer your question.
Yeah, thank you.
Yeah, I have another question regarding that sort of improving host chemical library. So you said previously library are essentially without any retention times. So you incorporate retention times into your library.
Yeah.
But actually retention time depends on many factors. You already mentioned it depends on the different conditions because retention time could be different.
Yeah.
So in other words, if somebody has generated data from other machines or whatever, it cannot use your library, right?
Yes, that's the reason why this is one of the issue when I didn't mention. When you're using the library, you have to stick with the same HLC, same solvent, the condition, because otherwise the rotation time changes.
That's right.
That's the reason why this is a tough question. And practical is hard. Is not every lab has that kind of resource able to make a library for themselves.
That's right. Yeah.
That's the problem. So, for example, I just show here, like the Ware Medical School facility director just told the machine, I just cannot do it because they just, so if Ware Medical School faculty, they're going to shipping their sample to us. So we will do here because we are just lucky we get some additional free gift able to make in those the library, and that's
great.
Yeah, and then that's why you really cannot using somebody else's library and using for us because anything column is also important. Different type of column, rotation time shift. So so that's always a very, very tough one. But Cornell, we're lucky. We're trying to keep. Well, one thing is that I want to send the message out is we're trying to keep building up more and more. So as I said, as later on, we can do retrospective research. So even your data generated now, I mean, 2 years from now, once we add more library, we can still do research, you get a benefit. So
what do you generally use for internal standards? Are you using like one internal standard based on the class of compound, or are you spiking in a mixture of internal standards?
Therefore, you're talking about for untargeted metabolomics.
For untargeted metabolomics.
Okay, so that's a good question. So for regarding internal standard, okay, so that's the also field. I'm talking field consensus. So this is the way how field are doing. For untargeted metabolomics, we using two different types, what we call standard, also quality control. One type of standard, like you said, we normally using, I think it's 7, right? More than 7. It's just different type of standard. However, that standard only using for our called QA, quality assurance, and you just make sure our system, every maybe every bench we start running, we make sure our machine everything is okay. So the really using the what we call QC, quality control, to answer your question, is we're going to using the mixture of each of the sample as the quality, what's called a QC sample. And there's 2 ways. One way is called a global QC. That's if you have a 40 sample like Justin here, that's what we did. If you have a 40 sample, we mix it all 40 equally together. That's called a global QC. The second QC is called a global QC. Global QC is we have 8 group, like 5 sample, we mix it together called a global QC. For global all together QC, we normally using for normalization of the quantitation profiling. So what that means, this is the reason why we need a QC sample. We actually running every 5 sample, we're running QC, the sample. And then we're using QC order on the intensity to normalize. The software allowed us to normalize back. And the reason why, right, the machine, when you're running day 1 on the first sample versus the day 3, the last sample, the machine response factor or sensitivity is going to drop. And the curve is like this. It start to drop. And then if you don't normalize, you introduce artificial, right? The quantitation change is not count. Obviously, we're using random running. That will really hurt your statistics. But after normalize, then everything's bring back. And then I want to add one more. Why we need a global QC? Because global QC, we're using identification or annotation. Because this group versus other group, wild type versus mutant, could be compound very different. So we're using both. Separated, the identification rate would be a lot of high. So that's what we do. So this is good. And again, we're always using the standard compound as a QA, quality assurance. In our system is fine. And the QC really using the normalized, make sure the data is going to be brought back to everybody is, basically same level. So that's what we use.
So you're just normalizing to your full QC.
That's right. Yeah, that's right. That's actually. And then again, QA sample is only for checking. Yeah.
Okay,
thank you for the
great.
question.
Thank you so much, Dr. Zhang.
This has been a production of Cornell University, on the web at cornell.edu.